Hi, my name is Mihai Pop, and I'm an associate professor in the Department of Computer Science and the Center for Bioinformatics and Computational Biology at the University of Maryland. Today, I will introduce you to the key concepts underlying genome assembly algorithms. To understand why genome assembly is necessary, I will briefly describe the shotgun sequencing process. The approach is to read the DNA sequence comprising the genomes of organisms. The squiggle on the slide represents the piece of DNA, say a chromosome, that we want to reconstruct. The sequencing process starts by duplicating the DNA into multiple copies. These copies are then randomly shared to generate a collection of random fragments of DNA. In general, the fragments are of roughly equal size, and the reason for this will become apparent later. The resulting fragments are then sequenced. That is, their DNA sequence is read by a specialized instrument called a sequencer. The blue segments on the slide correspond to the portions of the original fragments whose sequence is now known. As should be apparent by now, the sequence of the original genome cannot be determined without gluing together the individual pieces whose sequence was determined. This process is called genome or sequence assembly. To introduce another bit of terminology, the small DNA sequences generated by the sequencing instrument are called reads. By now you must be wondering why we're not just sequencing the genome rather than following this complex process. The answer is on the table you see on the screen. This table gives you a timeline of the development of sequencing technologies over the past almost 40 years. For almost 30 years, a single technology was widely used, Sanger sequencing. Starting with 2005, we have experienced a true revolution in terms of sequencing technologies, with a new technology becoming commercially available every few years. Pay attention, however, to the third column of the table. Despite the amazing developments made over the past 40 years, current sequencing technologies cannot read more than several thousands of base pairs at a time, while the genomes of most organisms range from millions to billions of base pairs. The shotgun sequencing process I have showed you on the previous slide is thus the only effective way through which we can reconstruct a genome, and assembly is a necessary part of this process. To recap, sequencing technologies can only read small fragments of an organism's genome, and a computational process called sequence assembly is necessary to put these fragments together into a single sequence. We are thus faced with the challenging task of reconstructing a gigantic jigsaw puzzle comprising from tens to thousands to tens of millions of pieces. To get a better idea of the challenge faced by genome assemblers, imagine that the whole shipment of a new book inadvertently fell into a shredder. Our task is to take the many small shreds and glue them together to reconstruct the original text. Assume, of course, that none of the copies of this volume survived the shredding process. Let us assume that the book we're talking about is A Tale of Two Cities by Charles Dickens. The first sentence in the book is shown on the screen. Now imagine we shred it into pieces that are exactly 4 words long. The recognizable sentence is now anything but recognizable, and it is not even clear that we can put it back together. We will try nonetheless. Let us pick a first read, coincidentally the actual first piece of the genome. It was the best. There are many other pieces that overlap this one, sharing anywhere between 1 and 3 words with the first chosen fragment. For now we will pick the sequence that most closely resembles the first one, that is, the read was the best of. Following the same strategy, we add the best of times and best of times it, and of times it was, slowly reconstructing Dickens' text. What we have just described is an assembly algorithm called the greedy algorithm. Greedy refers to the fact that this algorithm always makes the locally optimal choice by picking the next best overlap. Briefly, the greedy algorithm starts by examining all the reads to identify all the possible overlaps or shared sequences between them. Then it processes these overlaps in decreasing order of their length, iteratively joining reads together. Through this process, increasingly longer segments of the genome are reconstructed. The process ends when either no more overlaps are left, or all of the available overlaps conflict with already constructed segments. A simple example is shown in the figure, with the overlaps between 4 reads shown on the left and the sequence of joins outlined on the right. This very simple algorithm formed the basis for many popular assemblers, including FRAP, the workhorse of the public effort to sequence the human genome, and Tiger Assembler, the assembler used to reconstruct the first genome of a living organism, the genome of Haemophilus influenzae, sequenced at the Institute for Genomic Research in Rockville, Maryland in 1995. While I have shown you an approach for assembling a genome and told you that this approach has been quite successful, it is very important to keep in mind that as these technologies were being developed, it was by no means obvious that the assembly of shotgun sequence data was even possible. Scientists wondered if the randomness of the shotgun sequencing process may prevent us from ever reading every base in a genome. Even if we could prove that eventually every base would be covered, would we need to generate an infeasibly large set of reads to achieve this guarantee? And in the end, would computers be able to put all the data together? To clarify the first 2 points, see the figure on the screen representing the result of a hypothetical shotgun sequencing process. Each of the thin lines is a DNA fragment obtained from the genome of interest. Note that due to the random process through which the DNA is shared, some sections of the genome are better sampled than others. The plot on top indicates how many DNA fragments overlap at each position in the genome. You can now easily imagine that as we generate more and more DNA fragments, more and more of them will overlap until the entire genome is covered. A useful metaphor for this random process is watching raindrops on a sidewalk as rain starts to fall. As more and more of the sidewalk gets wet, dry spots persist for quite a while. We can similarly expect that gaps will remain in the coverage of the genome even as we sequence more and more fragments. But for how long? Eric Lander and Michael Waterman were the first scientists to explore the statistical properties of the shotgun sequencing process. They proposed a very simple model in which all reads had the same length and in which the reads had to overlap by some minimum length in order for the overlap to be recognizable by an assembler. They also took into account the total size of the genome and the number of reads produced. Using Poisson statistics, they were able to derive equations that describe the number and size of islands that one expects to find across the genome after a certain amount of sequencing. An island is simply a group of one or more reads that does not sufficiently overlap with any other reads. I will not go over the details of the equations, but I have plotted them for a range of sequencing depths. The sequencing depth, also referred to as coverage, represents the ratio between the total amount of sequence in the set of reads and the total length of the genome. As you can see from the bottom figure, as we increase the coverage from 0, the number of islands or contigs increases. Each DNA fragment is generated from a completely different part of the genome and does not overlap any of the other fragments. At a point, just past one-fold coverage, the number of contigs plateaus and starts to decrease. At this point, new fragments overlap existing fragments and sometimes even join existing contigs, thereby decreasing their number and increasing contig size, as seen in the top figure. As you can see, by about 8 to 10-fold coverage, the number of contigs approaches 1. That is, the whole genome will likely be fully covered by reads after sequencing about 8 to 10 times more DNA than the length of the original genome. These equations does not only prove that assembly is feasible, they also give us an idea of how much sequencing is necessary to generate assemblable data. We are still left with the question of whether the resulting data can actually be reconstructed computationally. Before we address this question, we need to more formally define the goal of the assembly process. One such definition equates genome assembly to the shortest common superstring problem. Specifically, for a set of strings sigma, we want to find a string S such that all the strings in sigma are part of S, and we want to make sure that S is the shortest such string possible. In the drawings on the screen, the bold strings both contain all of the 4 DNA stop strings, but the string on the left is shorter than the one on the right. This formulation of assembly assumes that the correct genome is the most parsimonious explanation for all the reads, assumption that we will revisit again later. Under this formulation, scientists have shown that genome assembly is an intractable, or NP-hard problem. This is a category of computational problem whose answer can only be found by sifting through an exponential number of solutions. In other words, assembly is impossible. Despite this negative result, computer scientists were able to show that simple algorithms such as greedy can give results that are not too bad. The greedy algorithm itself was shown to produce a string that is most 4 times as long as the shortest possible string, and it is conjectured that it may actually be at most twice as long. Let us return to the example I showed you earlier when we came up with the greedy algorithm. Let us add another read to the reconstruction of our genome, picking a read that overlaps the prior one the best, specifically by the string "it was the." We can very quickly find the string "it was the age" as a potential continuation of the assembly, but also "it was the best" and also "it was the worst." Which one should we choose? This example highlights a major drawback of the greedy algorithm. Since all decisions it makes are local, it can get stuck, leading to incomplete or incorrect assemblies. It turns out that the Achilles heel of the greedy algorithm are genomic repeats, segments of DNA repeated in identical or near-identical form throughout the genome. Here, I show you 2 simple examples. The first is an instance of a tandem repeat, multiple copies of the same string of DNA repeated multiple times, one after the other. On the left, you can see the correct structure of this tandem repeat, together with 7 reads generated from this section of the genome. It shouldn't be hard to see that the greedy algorithm will reconstruct the incorrect string shown on the right. Furthermore, this example also highlights that our formulation of assembly as an instance of the shortest common superstring problem is incorrect, as the string on the right is clearly the shortest common superstring. Also, note that while the example I'm giving you appears contrived, by replacing each A with a string TTAGG, you obtain a sequenced pattern commonly found in the telomeric regions of the human genome. On the bottom of the screen, you see another type of repeat with 2 copies that occur apart from each other in the genome. Again, the greedy algorithm can fail, this time by entering the repeat region from one end and exiting through the second copy of the repeat on the other end. As a result, the two copies of the repeat get collapsed into one, and the region between them remains in a separate config. To address the challenges posed by repeats, scientists have developed new approaches for modeling the assembly problem. These approaches rely on the computational formalism of graphs, or networks. An example is shown on the screen for our reconstruction of Dickens' work. The individual reads are connected to each other by an edge if they overlap. The graph clearly indicates the ambiguity that led us to be stuck earlier. The read times it was the is followed by either it was the worst or it was the age. Unlike the case of the greedy assembler, the global picture that emerges allows us to resolve this ambiguity in the following way. Assuming that we choose to take the path from times it was the to it was the age. Doing so disconnects the loop from the rest of the graph, leaving the text it was the worst of times it was in a separate config. Since our goal is to reconstruct a single config that represents the tale of two cities, we need to first follow it was the worst, and only after completing this loop continue with it was the age. Note that we could potentially traverse the loop multiple times, thereby generating a string of tandem repeats. In the case of Dickens, it is obvious that one traversal is sufficient, as the text it was the best of times, it was the best of times, it was the best of times would have surely prevented the book from being published. When dealing with biological data, such arguments cannot be made, thereby further complicating assembly. The ability to perform such holistic analysis of the assembly has led to the use of graph-based approaches in most modern assemblers. It is important to note, however, that repeats continue to be a problem. The example I showed you on the previous slide intentionally omitted information. The actual graph an assembler may see would look closer to what you see on the screen, where multiple traversals of the graph are possible yet indistinguishable from each other. In other words, given the same set of reads, we could reconstruct multiple genomes, all of which look equally good, yet only one of which is the correct one. In the case of Dickens, even knowledge of the English language cannot help us, as the various strings we can reconstruct, such as it was the worst of times, it was the age of foolishness, it was the best of times, it was the age of wisdom, appear grammatically correct. To further stress this point, here is the perfect assembly graph of the genome of Mycoplasma genitalium, a small 1 mega base pair long genome. This graph is constructed from short 25 base pair reads, and the different colors indicate the number of copies of each of the nodes in the graph. Despite the fact that there are no repeats with more than 4 copies, the red nodes, it is impossible to figure out how to traverse this graph and reconstruct the one circular genome of the Mycoplasma genitalium genome. There are 3 main paradigms for representing genome assembly as a graph problem. The first, termed overlap layout consensus, constructs a graph where the nodes are individual reads and the edges represent the fact that 2 reads overlap. The string graph is an extension of the overlap layout consensus approach, where some edges have been removed, as will be shown in the following slides. Finally, the Debrin graph approach, which has been very popular for the assembly of short read sequencing data, attempts to encode the repeat structure of the genome. The nodes and edges in this graph are short fragments of length k from the genome, also called k-mers. The reads are implicitly encoded as paths to the resulting graphs. The simple example on the screen demonstrates the basic structure of an overlap layout consensus graph. The individual reads are encoded as nodes in this graph, and the edges indicate that the corresponding reads overlap. Take, for example, reads 1 and 3. They share the string AGTG, and hence the corresponding nodes are connected by an edge. In this model, the assembly can be formulated as finding a traversal of the graph that visits every node exactly once, under the assumption that each read in the input must be used in the assembly. In the picture on the screen, the only traversal satisfying this property is the straight line connecting all the nodes, and you can easily see that this traversal correctly reconstructs the original string. Note, however, the tangle of unnecessary edges added to the graph. These, as well as repeats, can complicate the graph, making it difficult or impossible to find the correct path. Also note that the structure of the graph is intimately tied to our definition of an overlap. In our example, we require that at least 4 nucleotides match between adjacent reads. Increasing this parameter would eliminate spurious edges, but may also eliminate useful information. Decreasing the minimum allowable overlap would increase the number of spurious edges in the graph, possibly complicating the job of the assembler. A fairly simple extension of the overlap layout consensus graph was proposed by Gene Myers, leading to a new type of graph, the string graph. In a string graph, the graph structure is simplified by removing transitive edges, a process that removes many of the spurious edges seen in our example of the string graph. By transitive edges, we mean edges that encode information that can already be obtained from the graph. For example, the edge between reads 1 and 3 is spurious, as we can reach read 3 from 1 by following the path through node 2. The simplified graph for our example is considerably simpler, and the correct assembly is immediately obvious. In practice, however, considerable ambiguity in the graph structure remains due to repeats and sequencing errors. Note that the byproduct of the string graph transformation is that the overlaps encoded in the final graph are generally longer than those encoded by the edges that were removed. Again, looking at the first three reads 1 and 2 and 2 and 3 overlap by 7 characters each, as opposed to a 4-character overlap encoded by the transitive edge that was removed between nodes 1 and 3. The De-Bruijn graph approach focuses not on the reads themselves, but on what the reads can tell us about the sequence of the original genome. Each read is broken up into smaller substrings of length k, also called k-mers. The k-mers will end up representing the nodes in the De-Bruijn graph. Edges are created whenever two k-mers are immediately adjacent to the read, that is, the k-mers overlap by exactly k-1 letters. In the example shown on the screen, the first read will be broken up into GA-CA-CTA, ACA-CTA-G, and so on, and a similar process will be applied to the second read and the third, and so on. On the right of the slide, you can see the resulting graph. Note that my description above did not mention anything about overlapping reads, yet the graph constructed here clearly spans the entire sequence being assembled. The reason are k-mers shared by 2 or more reads, such as the string ACTA-GTG, which represents the overlap between the first and second reads. Note that the reads themselves do not get explicitly represented, rather they occur as paths through the graph. In the De-Bruijn graph, k-mers shared by multiple reads are encoded only once, irrespective of the depth of coverage of the genome, leading to a theoretical advantage over overlap layout consensus-based approaches where each read is explicitly modeled. The Bruijn graph approaches are, however, very sensitive to sequencing errors, which lead to the creation of false k-mers which complicate the graph structure and reduce its apparent efficiency. The development of competing approaches for representing genome assembly as a graph problem has led to considerable debate in the community over the relative merits of these approaches. The overlap layout consensus approach, for example, can be shown to be equivalent to a classic problem from computer science called the Hamiltonian path problem. This problem requires finding a path through a graph that visits every node in the graph exactly once and is known to be intractable. That is, solving it correctly requires one to search through an exponential number of possible solutions. The De-Bruijn graph assembly problem, however, can be posed as another classic computer science problem, the Eulerian path problem, or its extension called the Chinese postman problem. The Eulerian path problem involves finding a path that visits every edge of a graph exactly once, or at least once in the case of the Chinese postman problem. And efficient algorithms exist for solving these problems. This apparent advantage of the De-Bruijn graph approach is negated by the fact that for a typical graph there are an exponential number of possible Eulerian or Chinese postman paths, and thus finding the one path that represents the genome being assembled also leads to an intractable computational problem. In other words, genome assembly is computationally challenging irrespective of how we formulate it. Further complicating the choice between the competing approaches are practical considerations such as the challenges imposed by sequencing errors in the De-Bruijn graph approach and the relatively complex process needed to construct and analyze overlap or string graphs. As I have described the assembly so far, I kept referring to the assembly problem as computationally intractable. This statement can obviously be disproved by practical examples, the tens of thousands of genomes that have already been reconstructed through shotgun sequencing and assembly. To better understand this discrepancy between theory and practice, it is worth thinking of the whole genome sequencing process in terms of the information lost during this process. In very simplistic terms, we can imagine that the total information contained in a living organism is repeatedly depleted throughout the entire process, both during the experimental stages, like DNA extraction, DNA shearing, and so on, and the computational stages, such as assembly. Intuitively, shearing DNA into small fragments leads to a tremendous loss of information about the structure of the genome. But what exactly is this lost information, and how much information is really lost? The key insight lies in the relationship between the length of repeats and the length of reads. In the simple examples I have shown you when describing graph-based approaches, the correct assembly was immediately obvious. That was because in these cases the reads were longer than the longest genomic repeats. Our reads had length 10, while the longest repeated sequence was 2 base pairs in length. On the other extreme, however, when the reads are shorter than the repeats, the assembly problem is under constraint. There are an exponential number of genomes that could be constructed from the same set of reads. The computationally intractable instances of the assembly problem only occur when reads are roughly equal in length to repeats, a situation that rarely occurs in practice. At the same time, however, we frequently encounter the situation where reads are shorter than repeats due to the short length of the reads generated by modern sequencing technologies. This situation explains why, in most cases, genome assemblers cannot fully reconstruct a genome. Rather, they generate a collection of contigs corresponding to the segments of the genome that can be unambiguously reconstructed. To further demonstrate the relationship between the complexity of assembly and read length, on the screen you can see the assembly graph of the E. coli K12 genome sequenced with reads of increasing length. The graphs constructed from 50 and even 1,000 base pair reads are fairly complex, and one cannot easily find a path that spells the circular genome of E. coli. At 5,000 base pairs, however, the answer is obvious, as the graph is a single node and a single cycle. My presentation so far has talked about assembly in terms of reads alone. Can sequencing experiment provide us with more information? In many cases, the answer is yes. The shotgun sequencing process breaks the genome into smaller fragments which are then sequenced. For many sequencing technologies, it is possible to sequence both ends of a fragment, thereby providing information linking the resulting reads. Specifically, if the size of the fragments generated by the shotgun sequencing process is well controlled, we can infer how far apart the paired reads are in the genome. Before discussing how one can use this information in assembly, I want to mention that obtaining mate pair information can be difficult or even impossible, depending on the sequencing technology being used or the size of the fragments. The longest sizes achievable range in the tens to low hundreds of kilobase pairs, and the routinely achievable mate pair sizes are much shorter, ranging in the hundreds to low thousands of base pairs. As I already mentioned, genome assemblers usually generate a collection of contigs rather than a single one per chromosome, due to either complex repeats that cannot be resolved, or due to regions of DNA that are simply missing from the sequencing data. Mate pair information can help link together these contigs, thereby clarifying their relative location in the genome. Specifically, if one of the reads belongs to one contig and its pair belongs to another one, we can infer that the two contigs occur next to each other in the genome, and we can estimate the distance between the contigs based on the approximately known size of the mate pair. This process is usually called scaffolding, and it results in a more global representation of an organism's genome consisting of a string of contigs separated by gaps with unknown sequence but of an approximately known length. Mate pairs can also help resolve genomic repeats that could not be disambiguated by the reads alone. Specifically, the approximately known distance separating the ends of a mate pair can help constrain the possible traversals of the assembly graph. In the figure on the screen, you see a small part of an assembly graph where two repeats, the blue ovals, confuse the reconstruction. Assume that a mate pair connects the filled nodes and that we know the approximate size of this mate pair. Note that there are two ways to traverse the graph between the filled nodes, one going through the small circular node between the repeats and the other going through the oval node. These two paths have different lengths, and in our case only one of them is consistent with the known separation between the filled nodes. This is the thickened path passing through the circular node. In addition to methods aimed at resolving repeats, genome assemblers rely on approaches to identify and remove repeats in order to simplify the assembly problem. One of the commonly used approaches involves looking for regions in the assembly that are too deeply covered. One can phrase these questions as a statistical test, and a number of such tests have been developed in the community. A popular one, exemplified on this slide, is the A statistic from Celera Assembler. The A statistic is simply the log ratio of the probabilities of a particular genomic region having been generated from a two-copy repeat versus unique DNA. The probabilities are derived from Poisson statistics. Coverage-based approaches do not work in situations where the sequencing depth of coverage is highly uneven, such as in metagenomics or single-cell experiments. In such cases, graph-based approaches can be used, aimed at identifying contigs that tangle the assembly graph, such as the gray node on the screen. This strategy was initially proposed in a Bambus 2 scaffolder, a tool developed for metagenomic analysis. To evaluate how useful mate pairs are for resolving repeats, we simulated mate pairs with different sizes and estimated how much of the repeat-induced ambiguity is resolved by these mate pairs. The results of this experiment are shown on the screen. The scale on the y-axis roughly corresponds to how much of the genome is resolved by repeats, with 100% indicated the genome can be reconstructed into a single contig. There are two observations we can make from this figure. First of all, no matter how many mate pairs we generate, we cannot fully resolve this genome. In other words, mate pairs are not a foolproof solution to the assembly problem. Second, the shorter mate pairs, in this case about 300 base pairs in length, led to the most improvement in the assembly. This is a somewhat counterintuitive result, as we can expect that the longer mate pairs can span longer repeats and thus be more useful. At the same time, extremely long mate pairs are not very useful. Imagine, for example, mate pairs connecting the telomeres of human chromosomes. These do not tell us anything about the actual structure of the chromosome in between. Clearly, there is a sweet spot for the size of mate pairs in terms of how useful they are for assembly. To understand what is going on, see the graph shown on the screen. Unlike the previous slide, the two nodes in between the repeats are the same size. As a result, the mate pair connecting the two red nodes is useless in resolving the repeat, as the two alternate paths both have the same length. At the same time, the shorter mate pair connecting the two green nodes can help the assembler by indicating that the correct reversal of the graph, starting from the leftmost green node, must pass through the second green node rather than the white one above it. To summarize, the mate pairs are only useful for assembly if they match the repeat structure of the genome. To further demonstrate this point, we used a collection of about 400 bacterial genomes and simulated, for each of them, two mate pair strategies. The first was a standard mixture of two libraries, one of about 1,000 base pairs and the other of about 10,000 base pairs. The second strategy involved tuning the mate pair libraries for each genome by picking the two mate pair libraries most likely to resolve repeats. In practice, these libraries were much shorter, in the range of several hundreds of base pairs. On the screen you can see the results of this experiment. On the left are the results for standard mate pair libraries. The height of the bars indicate how much of the ambiguity was resolved, and the x-axis corresponds roughly to how difficult the genomes are. On the right are the results for the tuned mate pair libraries. As you can see, these libraries led to much better improvements in the assemblies. To summarize, the results I showed you stress the importance of the experimental design in obtaining good results. A common mantra in computer science is garbage in, garbage out. That is, the result produced by the assembler can only be as good as the data fed to it. Scientists must consider the requirements of the analysis algorithms before generating the data. A great example of this principle is the All-Pads-LG assembler from the Broad Institute. The authors of this tool specified the sequencing strategy that needs to be followed to generate the data. Datasets that do not meet the specified requirements are simply rejected. The strategy suggested by All-Pads-LG includes very short libraries where the mate pairs overlap. This information can be used to glue together the overlapping mates and effectively generate longer reads. As we've discussed already, longer reads lead to better assemblies. The All-Pads-LG strategy also includes mate pair libraries of increasing sizes aimed at capturing different classes of repeats or gaps in the assembly. Now that we know how to generate an assembly, how do we interpret and evaluate it? The simplest and most widely used measures are simple aggregate statistics related to the size and number of contexts generated. More sophisticated users may also care about how much of the input reads are contained in the assembled data. Large numbers of unassembled reads may indicate low-quality input data, sample contamination, or high levels of assembly errors. Aggregate statistics, such as averages, can be unduly affected by outliers such as tiny contexts. Thus, it is useful to also look at accumulation curves that represent how much of an assembly is contained in large contexts. An example for 3 different assemblies of the same data is shown on the slide. The contexts have been sorted by the size in reverse order along the x-axis, while the y-axis shows the total amount of DNA contained in contexts larger than the corresponding x-coordinate. Some trends are immediately apparent. The blue assembly, constructed with SOAP de novo, contains larger contexts than the other assemblies but comprises overall a smaller total size, while the black All-Pads-LG assembly has smaller contexts and a larger total size. The red Mazurka assembly has contexts smaller than either SOAP de novo or All-Pads-LG and comprises less total DNA than All-Pads-LG but more than SOAP de novo. Such accumulation curves are frequently summarized in a single value, the N50 size, corresponding to the x-coordinate or context size, where the accumulation curve crosses the red horizontal line representing half of the total genome size. By this measure, the blue assembly wins over the black and red ones. The main missing part from this assessment are errors. Context size is, at some level, irrelevant if it comes to at the cost of too many errors. There are many kinds of errors that can make their way in assemblies, such as misjoins or chimeric contexts, incorrect estimates of the consensus sequence, and missing information, such as gaps or fragmented contexts. Such errors are at some level due to the inherent complexity of assembly and imperfections in the data. It is important, however, not to forget another common source of errors: computer bugs. The picture on the screen shows the first computer bug, which was an actual bug that Grace Hopper found in 1947 in a relay of one of the early computers. Software engineers largely agreed that most software contains between 15 and 50 bugs per thousands of lines of code, extrapolating to thousands of possible software bugs in typical assembly software. Detecting assembly errors involved identifying internal inconsistencies in the output of the assembler. For example, the assembly shown on the screen has merged together 2 sets of reads, the green and the yellow, that differ from each other at several locations highlighted in purple. While such a picture would be expected in an assembly of a diploid organism, such polymorphisms may indicate assembly errors in the assembly of an isolated bacterial genome. Additional consistency constraints are provided by mate pair information. Several scenarios are shown on the screen with the correct assembly on the top and incorrect one on the bottom. The mate pairs represented with dashed lines are inconsistent with the expected size or orientation. Reads that do not fit well in the assembly can also signal problems. The example on the screen shows reads that all align to the assembly in an unusual way. Their first and second halves align to disjoint positions in the assembly. This signature is indicative of the presence of a tandem repeat that was collapsed into a single copy, thereby kicking out the reads that span the join between the two copies. The example shown on the screen is from an actual assembly of the Bacillus anthracis genome, and this misassembly led to the development of the diagnostic markers used to identify the origin of the strain used in the bioterror attacks of 2001. Assembly quality can also be evaluated in a more global way by estimating the consistency of the assembly with the parameters of the experimental protocol used to generate the data. In probabilistic terms, we can estimate the likelihood that a set of reads would have been generated from the assembly. Such holistic quality measures, which summarize the assembly in a single number, are particularly useful in comparing multiple assemblies of the same dataset and in automatically tuning the parameters of the assembly process. Before concluding the discussion on the validation of assemblies, I would like to note that this issue has been receiving a lot of attention in recent years, in part due to the fact that many competing assembly tools now exist in the community. Several competitions have been organized, the most prominent of which are Assemblathon and Gauge. The Assemblathon competition focused on the assembly of large diploid genomes using both artificial and real datasets. The Gauge competition looked at real data from genomes of varied sizes. Neither competition was able to identify a perfect assembler, rather depending on the dataset, different assemblers performed better. Before concluding, I would like to point out that the genome assembly research has a long history that goes back to the pioneering work from Esko Ukkonen's group in the mid-1980s. Many theoretical and practical issues have already been addressed by the pioneers in this field, though challenges and new opportunities remain, primarily due to the rapidly changing landscape of genomic technologies and the broadening application of sequencing technologies to new biological problems. In practical terms, the characteristics of the data being provided to an assembler are of paramount importance to the quality of the output. It is thus extremely important to pay close attention to issues such as experimental design and data cleanup. Despite repeated claims by the authors of assembly tools, no algorithmic paradigm or assembly software can best solve all the problems faced by biologists. It is critical that you carefully evaluate multiple tools and multiple sets of parameters to identify the strategy that best fits your data and the question you are trying to address. Finally, it is very important to thoroughly validate the output of genome assemblers, going deeper than simple aggregate statistics. Identifying and quantifying assembly errors can help not only choose a specific assembly strategy, but it can also help inform downstream analysis that rely on and assume the correctness of the assembled data. For more information about genome assembly, including key publications, links to assembly tools, as well as information about the active research in genome assembly at the CBCB, please follow the link shown on the screen. Thank you for your attention.